guys, and welcome back to Biologue. Hope you're having a great day and a great week. Now this week's video is going to be an IGCSE past paper video based on heart and circulation. So we're going to cover some questions on double circulation, single circulation, and how the heart pumps blood, what are the different heart chambers like the ventricles, the atria, etc. So we're going to cover these types of past paper questions in this video, and the next video is also going to be a past paper based video based on drugs. So we're going to look at recreational drugs and medicinal drugs. So make sure you stay tuned for that video. Unfortunately, these will be my last few IGCSE past paper videos, but I will still have biology based videos and exam revision tips and study method videos available for you guys. So make sure you stay tuned for that, and make sure you subscribe to Biologue and like this video for more such notifications and videos in future. And without further ado, let's begin. Alright, looking into the first question, amino acids are absorbed from the small intestine and transported to the liver. Describe how the liver is involved in metabolism of amino acids. So in this case, you need to talk about what exactly are amino acids and what are they used for. So amino acids are the substances which are used to make protein. So that's your first point. So amino acids are substances used to make proteins. Substances, you can use a more scientific term for it by saying a monomer. They are the monomers used to make proteins. Alright, used to make proteins. So that's your first point. Now, what exactly does the liver do, and what is its role in metabolism of amino acid? That's what the question is asking you, right? What does the liver do? The liver helps in deamination. The process name is called deamination. So the liver helps in the deamination of amino acids, which is the breakdown of the amino acids. Okay, it's the breakdown of the amino acids by removing, so breakdown of amino acids into urea. Okay, that's very important. So amino acids to urea by, how does it do it? By removing the nitrogen containing part or group of the amino acid, right? By removing the nitrogen containing group of the amino acid. Nitrogen containing group of the amino acid. And this is also called your amine group, amine. So if I had to explain what amine was, which is not really necessary for IG, but it's just to aid your explanation and just to aid your understanding. So the amine group, like for example, this is the structure of the amino acid. Here, this NH2 is called your amine group, all right? This NH2 is called your amine group. And here, where you have your COH, this is called your carboxyl, a carboxyl group, right? All right, this is the structure of the amino acid. And here you're wondering what this R is. This R is basically your R group, and your R group changes per amino acid, and that is what determines what type of amino acid it is. And so it determines, certain properties of the amino acid, like the structure and hence the function of the protein that is going to be found from the sequence of, a specific amino acid sequence, basically. Yeah. So all right, so that was the next point. Something else you can add here is that, so we have talked about deamination, what exactly it is, and we've talked about the formation of urea. You can also add that now, because the amino acid is made of this carboxyl group, you can say that this carboxyl group, or the carbohydrate part of the amino acid, can be used for production of energy in respiration. Okay, so the carboxyl group or carbohydrate group, anything is fine, carbohydrate group in the amino acid can be used for energy production in respiration. Can be used for energy production. It can be used for energy production in respiration. All right, moving on to the next question. So describe the effects on the liver of the following. Insulin release from the pancreas. So in this type of question, you need to talk about what exactly happens when insulin is secreted. So when insulin is secreted, it stimulates the liver. All right, what does it do? It stimulates the liver to do what? It stimulates the liver to absorb more glucose so that this glucose can be broken down or converted by the insulin into glycogen, so that the glucose can be, I'm just writing it in points again, but you guys need to write this in complete sentences, so that the insulin can convert glucose into glycogen. You can say convert or break down, but convert might be a better word to use here. So because it's not exactly breaking down, it's converting, right? So the insulin can convert the glucose into glycogen. All right, this is your 2 points for 2 marks. But if you want to add an additional point, you can say what exactly this glycogen will be used for, which is that it will be used for storage. Okay, storage in case of energy requirements that need to be met later. All right, moving on to the next question. So we have the second part talking about adrenaline being released from the adrenal glands. Now you have to talk about what exactly is the role of adrenaline. All right, what does adrenaline do? Adrenaline breaks down the glycogen into glucose, breaks down glycogen into glucose. Glycogen to glucose, that is your first point. All right, and this word breakdown is very important in this case because that's a key term. So you need to make sure you write that. All right, so it stimulates the liver cells. Yeah, you can say it stimulates the liver cells. That might be a better term. So you can say it stimulates the liver cells. It stimulates the liver cells to secrete adrenaline, which will help in the breakdown of glycogen to glucose. That's how you're supposed to structure it. So you can say that the adrenaline released from the adrenal glands stimulates the liver cells and helps it in the breakdown of glycogen into glucose. All right, moving on to the next question. Excessive long-term consumption of alcohol. So you have to talk about what exactly does alcohol do? All right, what are the effects of alcohol consumption? So alcohol consumption specifically on the liver, it can cause the accumulation of fat droplets, right? Accumulation of fat droplets or deposits, you can say fat droplets or deposits, and this accumulation of fat droplets or deposits can in turn cause a condition called cirrhosis, which is basically where your liver becomes quite full of a buildup of fat deposits. So the liver has a buildup of fat deposits. You can just write this in bracket because I mean technically you're repeating the same point, but it's just to aid your explanation. So this can cause cirrhosis, and cirrhosis can in turn in future lead to things like liver damage, severe liver damage, which can in turn cause liver failure. So liver damage, and you need to make sure that you write severe liver damage, which can cause liver failure. All right, liver failure. Okay, now moving on to the next question. So cholesterol can accumulate in the gallbladder to form gallstones. So the main cause is that cholesterol can lead to gallstones, and these gallstones will stop bile from flowing from the liver. Okay, bile flowing from the liver through the bile duct into the duodenum. Okay, now explain the possible effect of gallstone on the digestion of fat. So pretty much what exactly happens when you're not able to allow bile to move from the liver to the duodenum, right? Where is bile used? Bile is used in the small intestine, which is the duodenum. And here you have to talk about what exactly bile is. What does it contain specifically that helps in the process of digestion of fat, right? So when we look into that, we have to say that bile is something that contains bile salts, right? Bile contains bile salts. And what do these bile salts do? These bile salts help in the emulsification of fat. This term emulsification is very important. It's a key term, all right? So it helps in the emulsification of fat, and because it helps in the emulsification of fat, what exactly is it? It's the fact that it breaks down large globules of fat into smaller globules, breaks down larger globules of fat. And the term globules is very important. It's a very good term to use. So it breaks down larger globules of fat into smaller globules. All right, into smaller globules. And then what you have to say is that once it breaks it down, what happens? Now because bile does this process of emulsification, it increases the surface area of the fat for lipase to For lipase to catalyze the reaction, the breakdown. To catalyze the reaction or the breakdown of fat into fatty acid and glycerol, into fatty acids and glycerol. Okay, let's look into the next question. Now it says heart surgeons may stop the heart beating during operations, right? While this happens, blood is pumped through the heart using, a heart-lung machine that oxygenates the blood. So this particular machine, what it does is that it oxygenates the blood. Now figure 1.1 is a diagram showing the heart-lung machine in use. All right, so you have your figure here. Name the structures labeled A to D. That's the first question, A to D. So A, as you can see, is the left atrium, right? It's the first one. Always when you're doing these types of labeling questions, I would suggest just writing it like this. LA, LV, you have RA and RV, right? You have left atrium as the first one. Now B is between the left atrium and the left ventricle. So you have your bicuspid valve, okay? B is the bicuspid valve. C, what is C? Now this is interesting. C is actually your, so if you look at it, where is it present? That's the interesting part, right? Now this is in between two connections where you have one connection being the left ventricle and the other connection being the aorta. That's why here, F, we can already see that it is aorta, right? Why is it aorta? Because now it's bringing the blood, into the, or out of, yes, it's bringing the blood out of the left ventricle to the rest of the body, right? That's how we remember aorta, right? It brings the blood out of the left ventricle to the other parts of the body. Now, when we look at C, we need to understand that F is the aorta. So automatically the connection between the aorta and the left ventricle would be a semilunar valve. So C is a semilunar valve. A semilunar valve. All right. Now what exactly, where is the D? Right. D, already we have labeled it. It is the right ventricle, right? It's the right ventricle. And E must be the, all right, E, now it goes to the, right atrium. All right, so it goes into the right atrium, which is why it is the superior or inferior vena cava. All right, so let us now label the structures again. We have A, which is the left atrium. So left atrium. B is the bicuspid valve. B is the bicuspid valve. C is the semilunar valve, right? We labeled that already. C was the semilunar valve. And D we know is the superior or inferior, oh no, D was the right ventricle, right. So D was the right ventricle, which we have labeled over here, alright. So D was the right ventricle. Okay, let us look at the next one. Name the blood vessels E and F. So we've already done this, right? We know F is the aorta and we know E is the superior or inferior vena cava. Superior or inferior, because you don't exactly know which one it is. You need to write both. So it is the superior or inferior vena cava. So let us write that down. E is the aorta. Oh, sorry, 1 second. E is the superior or inferior vena cava. Okay. E is the superior or inferior vena cava and F is the aorta. Alright, moving to the next question. The heart-lung machine is used so that surgeons can operate on the arteries supplying the heart muscle, and these arteries may be diseased, okay? name these arteries and explain how they may become diseased. Now, for you guys, you only have learned about the coronary artery, which is a type of artery that can become diseased, right? With, you can have your fat lumps coming and then depositing there, and that can cause blockage. That's the only artery you've learned about which could potentially become diseased. So automatically your brain should think, okay, the name of the artery which can be diseased is the coronary artery, because that's the only one I have learned, right? As of now in IGCSE. So the name of the artery is your coronary artery, and the explanation is how exactly this happens. So what is the disease? What exactly happens? So the explanation is that there is a fatty deposit. So fat deposits in the wall of the artery, in the wall or the lining of the artery, and what exactly does this fat deposit do? It restricts the flow of the healthy blood, okay, which restricts the flow of the healthy blood, okay, and it can sometimes lead to clotting, and heavy fat deposits can lead to clotting, okay, heavy fat deposits can lead to blood clotting, and this blood clotting further restricts the flow of blood, which further blocks the flow of blood, further blocking the flow of blood, and hence it restricts the oxygen supply to the heart, because we know the coronary artery supplies the glucose and oxygen to the heart, right? So it restricts the oxygen supply to the heart. This is your answer. Moving on to the next one. Alright, so suggest why a patient is put on a heart-lung machine during such an operation. Why? Alright, the reason why they're put. This is because now the heart is actually not pumping blood. You need to look back at the question, right? What is the question saying? It says that, while this happens, blood is pumped through a heart-lung machine that oxygenates the blood. So here your answer is already given, right? Because your, heart is not able to pump blood since it sort of stops during the operation. The heart is unable to pump blood or is not pumping blood. Hence, you need to oxygenate it. Hence, it needs to be oxygenated. Or the CO2 needs to be removed. From the blood. This is why you're using a heart-lung machine. Alright, so this is your answer. Moving on to the last part of this question. And now humans have a double circulation system. Right, we know that. There is low pressure circulation and high pressure circulation. You have to explain how the structure of the heart helps to pump the blood in two circulations at two different pressures. So, number one, you're going to talk about, heart structure, right? So the heart, it consists of your ventricles and your atrium, right? Or atria. Consists of two separate ventricles. You can name it if you like. You can say left ventricle and right ventricle. Okay, left ventricle and right ventricle. Now, what exactly do these ventricles do? The ventricles, they pump the blood. Okay, ventricles pump the blood by contracting at high pressure. So when they contract, this creates an atmosphere of high pressure. This creates high pressure for the blood to flow. Okay, high pressure for the blood to be pumped. Alright, this is how the ventricles work. And how is it that the pressure now is sort of maintained or different for, the right ventricle and the left ventricle? This is because of the thickness of the muscle and the thickness of the wall. So you have to talk about the thickness of the wall, as in the right ventricle has a thinner wall. The right ventricle has a thinner wall or thinner muscle, and the left ventricle has a thicker wall. Left ventricle has a thicker wall or muscle. So the left ventricle is able to pump blood under high pressure to the rest of the organs in the body, whereas right ventricle does not need to pump it at, very high pressure. It can pump it at low pressure because now it's only going into the lungs. So it will still have some time for reoxygenation and for removal of the carbon dioxide. Right, so the right ventricle has a thinner wall, which is why it pumps the blood at low pressure. Okay, this happens to the lungs for oxygenation of blood, and this happens to the rest of the body organs. All right, so that is your answer. Blood flows through the hepatic portal vein from some organs to the liver. Figure 2.1 shows the hepatic portal veins and these organs. So you have the liver, you have the hepatic portal vein, and you have an organ, spleen here. Alright. Now, blood in the hepatic portal vein is deoxygenated. Explain why blood in the hepatic portal vein is deoxygenated rather than oxygenated. So here you're supposed to talk about how, capillaries, they build up and they join to form veins, and multiple veins join to form the bigger hepatic portal vein. Now, this blood that is actually passing through, the blood will pass through the capillaries which are present in the organs, right? So because of this, when the cells in that organ respire, they release carbon dioxide into the blood, right? So now, because of lesser concentration of oxygen compared to carbon dioxide, it becomes deoxygenated. So your main point here would be that the blood that is passing through, the blood is passing through capillaries, alright, and these capillaries are present in the body organs, alright, and so upon respiration of cells, the blood becomes deoxygenated. Upon respiration of cells, the blood becomes deoxygenated. Okay, upon the respiration of cells, the blood that actually passes through the capillaries, it becomes deoxygenated, right? Because it has been through a specific organ, organ. So the cells in that organ would have lost oxygen, or would have sort of respired, right? So cells, due to respiration, it would have released carbon dioxide, okay, CO2, and the cells actually, because now from the environment, alright, you're taking in oxygen, so the oxygen from the blood would have been lost to these cells, You can write any 2 points because it's actually 2 marks here. You can write any 2 points from these, but definitely make sure that you're reading the question well, because this can be a tricky question if you don't think about it, alright? Explain how the liver is involved in regulating the composition of blood and in protecting the body against toxic substances. So you have to talk about the role of the liver in regulating blood composition. That is the first thing. So when you talk about that, remember that there is always glycogen. Alright, glycogen is kept as a store in the liver, right? It's kept in the form of storage in the liver. So that's the first thing that should come to your mind, that okay, glycogen is present in the liver. So what happens now in the liver when there is a low concentration of glucose or blood sugar, alright, the glycogen is converted into glucose, right? How glycogen is converted into glucose? How? By secreting glucagon. Now again, make sure you're writing this in full sentences. I'm just writing it in points to make it easier for you guys to understand. So remember that low glucose concentration means that glycogen gets converted into glucose, and this is released into the blood. So it helps to maintain the blood sugar concentration. You can say maintains the blood sugar concentration by releasing or secreting glucagon. Second thing, you can write the opposite of what's been said here, and you can talk about how there's high glucose concentration. So what happens when there's high glucose concentration, right? So when there is high glucose concentration, all right, the glucose gets stored and converted. Glucose gets converted to glycogen, and this glycogen is stored in the liver. So this actually decreases your glucose sugar concentration, decreases the blood sugar level or blood sugar concentration. So it helps to maintain the blood glucose level or blood sugar concentration. All right, this happens due to the secretion of insulin. All right, you have to write the name of these hormones. That's very important. And then the third point now, you've covered 2 points for this, right, the blood part. Now you can also talk about how typically, in the liver, right, we're talking about the liver, so you can talk about how toxic substances like alcohol, all right, toxic substances like alcohol are broken down or metabolized in the liver, are broken down or metabolized in the liver, all right, and this helps to maintain the composition of the blood, hence regulating. This is how you'll end your answer, hence regulating the composition of the blood. Alright, so here these are your points. You have number 1, the low concentration you've talked about, that's 1. You have glycogen to glucose 2. Here 3, mentioning of glucagon. Then there's 4, when high glucose concentration is there, so that's glucose to glycogen. Then the mentioning of insulin is 5, and this toxic substances point is 6. So you always make sure that you have more points written than the number that they've asked you for. Like here there's 5 points that's being asked from you and expected from you, but you should make sure you write at least 6, so that in case you forget one of the points related to this, you have one more point in hand that you can write. That'll be it for today's video, guys. I hope you liked it. If you did, make sure to like, share and subscribe to Biologue for more such videos, and make sure that you put your post notifications on as well. And I will see you guys in my next video. Have a great day.